when working with microorganisms, doing either just research or also treatment, we do consider different biosafety levels. So these are recognized levels that have been established, and there are set conditions and rules that must be followed when working with the different microorganisms in these categories. If you look at the diagram, the triangle there at the top of the slide, you can see that biosafety level number 1, that is the lowest risk microbes. And then as we move up, the microbes get more and more dangerous. All the way up to biosafety level number 4 would be the highest risk microbes as far as what impact they might have on us. So if we look in closer detail at these different biosafety levels, first of all, biosafety level number 1, again, that's the bottom of that triangle, that is going to be the lowest risk microbes. So these are going to be conditions that are designed for microbes that are not known to cause any particular human diseases. When we're talking about biosafety level number 2, then it's a little bit more dangerous. So this would be designed for microbes that are moderately hazardous. If we get to biosafety level number 3, then we're getting into much more dangerous things. So this is going to require that work be done in safety cabinets in many cases. This will be safety cabinets that are equipped with special filters, which we call HEPA filters. Those HEPA filters have really small holes or pores, which are going to be capable of filtering out most of the microbes. So we have special hoods that individuals would work in, certainly different gloves, and the use of special ventilation. And again, this would be to protect both the researchers that are involved in that, and then ultimately to protect the general public. If we look at biosafety level number 4, this is going to be the most dangerous, the most hazardous of all of the organisms. So these safety level conditions are designed for working with dangerous or exotic microbes that cause very serious or fatal diseases. This is going to require the use of isolated buildings. There's going to be very strict entry and exit. You would have to have special approval to be able to enter and exit from those buildings, and they require airlocks. It would be what you see in this picture right here, where individuals have to wear special kind of space suits that are going to be airtight, that basically prevent them from inhaling or touching or coming into contact with that microbe that they're working with in any way. All right, if we talk about physical methods of microbial control, there are a number of physical ways that we do attempt to get rid of microbes. One of the most common is going to be, first off, using high temperatures. High temperatures are very effective at, first of all, denaturing proteins, which will be inactivating those proteins by unfolding them. These temperatures are also going to interfere with the membranes of the cell, also the cell wall integrity, and also disrupting or deactivating the nucleic acid structure and function. So all of those things would potentially kill microbes, cause them to not be able to reproduce, probably not be able to perform any metabolic functions as well. The thermal death point is the lowest temperature that can actually kill all the cells in a particular sample within 10 minutes. So this is something that is investigated to determine what the thermal death point for a particular microbe would be. The thermal death time is described as the amount of time that's required to completely sterilize a particular volume of liquid at a set temperature. So this would be microbes in solution, and we've got a specific temperature, so we want to know how long will we actually have to heat them for. Now, if we were to plot this and look at a graphical representation of this, this is showing us what we refer to as the decimal reduction time. The decimal reduction time is a constant value, and this is going to be the time that's required to destroy 90% of the microbes in a particular sample. Now, if you look on the y-axis, it is a logarithmic scale, and basically every time we go down basically tenfold, that would be destroying 90% of the organisms there. So you can see right here that it's showing us that in that decrease right there, that's 90% that would be destroyed. So for this particular example, what it's showing us is basically it takes about 2.5 minutes roughly to kill off 90% of the organisms. So this would be the decimal reduction time for this particular microbe that we're looking at here. When we're talking about utilizing temperature, there is a difference between using dry heat and moist heat. Moist heat is always going to be more effective, and that is because water is a really good conductor of heat. So if we're talking about boiling, that would be an example of using moist heat. This is very effective for sanitizing, but it's not real effective for true sterilization. So again, true sterilization means we are killing absolutely everything. But sanitizing, we're getting rid of the harmful ones, the pathogenic ones, but there's still going to be some microbes left. This is something that is very commonly used when we're in different restaurants. So in restaurant conditions, we're many times sanitizing the dishes, sanitizing the eating utensils using very, very hot water. Many of the dishwashers, washing machines that we have these days, they are able to reach very high temperatures and sanitize the dishes or sanitize the clothing. So again, getting rid of the pathogenic microbes. Now, one of the reasons why this is not going to be good for true sterilization is because boiling water has a set temperature. Boiling water at normal atmospheric pressure is going to be at 100 degrees Celsius. And so we can't really get higher than 100 degrees Celsius when we're boiling. So if we want to get a higher temperature than 100 degrees Celsius, what we have to do is we actually have to increase the pressure. So you can see normal atmospheric pressure is right down here at 0, and that's where we have 100 degrees Celsius. So under normal boiling conditions, 100 degrees is as hot as we can get. So if we want to get to a higher temperature, that is where an autoclave becomes very useful. Because what an autoclave will do is it will actually use some pressure at the same time, and that is going to have pressure. We're also going to have boiling water, and because the steam can't get out, the temperature is going to be much higher inside of there. So we're basically sterilizing now with high temperature and high pressure. And this is also going to be moist heat that we're using because it's going to be the water, the boiling water, the steam that's going to be doing the sterilizing. So here's just a quick look at an autoclave. An autoclave really, when you get down to it, this is like a pressure cooker, but it's going to be larger in many cases. This is one way that they sterilize many instruments when we're talking about a medical environment. So the instruments are put on a tray perhaps, and then it's inserted in there. There is typically going to be some water in the bottom of the autoclave so that when we close the door and we tighten it up so that it's airtight, that water can be boiled, and then that boiling water under the pressure is going to reach a very high temperature, and then we're going to basically do that for maybe 10 minutes, 20 minutes, really depends on the particular microbes that we're trying to eliminate. The volume of the liquid is also going to be important as far as the actual amount of time that we're going to be sterilizing for. Pasteurization is an example of using some moist heat. This is going to be when we heat a solution just enough to destroy spoilage microorganisms without affecting the taste. We don't want to heat too much because when you do start to heat to too high of temperatures, many of the things that give a product its flavor, they will start to break down. So that would definitely not be something that we want to do. We want to avoid breaking down and affecting the taste or the flavor, but we do want to get rid of the organisms that are going to spoil it. So we go just as hot as we have to get rid of those spoilage microbes. Ultra high temperature sterilization is going to basically take the liquids, it's going to heat them up really fast, and then it's going to cool them off really fast. So when we're talking about this, we are passing liquids through superheated steam. That's typically around 140 degrees Celsius, but we're just doing it for a few seconds. And again, the reason for this is that we don't want to break down the product that we're trying to sterilize. And then we do cool it off very fast. If we talk about dry heat is going to use hot air. And so the hot air, it is high temperature, so it can certainly denature proteins. It can foster oxidation of various chemicals. This is, though, going to require much higher temperatures than when we're using moist heat. So an example of the way that we might treat something when we're talking about dry heat is that we might heat it up to maybe 171 degrees Celsius for an hour or so. So notice that it is going to require much longer as far as the overall treatment goes, and that's because it just takes longer to conduct that overall temperature. Refrigeration and freezing can also be used for controlling microbe growth. And the main reason why that's going to be the case is because reactions overall occur much more slowly at low temperatures. That's one of the reasons why our food doesn't spoil as fast when we do put it in the refrigerator, because reactions that microbes would be performing are going to occur much more slowly. Another thing that happens is that when the temperatures get really cool, the liquid water becomes limited. It's not in as great of a supply because much of it is freezing, and liquid water is needed for many chemical reactions. This overall is going to decrease microbe metabolism, it's going to affect growth, and it's also going to decrease the overall reproduction rate. Desiccation is drying. So desiccation would be drying that limits the overall availability of liquid water. Again, this is going to limit a lot of microbial reactions, the metabolism that takes place, the reproduction, because water is a very important component of that overall metabolism process. If we take cooling and we take desiccation and we put the two together, this is going to be what we call lyophilization. So we're combining freezing and drying, and this is very often going to be used for preservation of microbes. If we want to store microbes long term, they can be lyophilized, and in this case, they can be stored for decades. And then when we want to grow them again, they can be taken out of the particular freezer that they're in, and then we can rehydrate them, warm them up, and we can actually kind of wake them up at the same time and then culture those microbes. Filtration is going to be a way to remove microbes by basically filtering them through some type of sieve that has really small pores, really small holes. So if we look here, we're going to have some type of filter in between these two containers that you see here, and we're going to pass the fluid through this sieve, which has very small pores, and this is going to trap and separate out the particles. If we look at this image right here, this is showing us a look of this at the sieve. These black spots, those are the pores, and anything that's larger than that pore is going to get trapped. So that's all the purple cells that we see here. They are not going to be able to pass through this particular filter. This is pretty good at getting rid of a lot of different microbes. Sometimes you do have some really tiny ones that would squeeze through and make it into the sample at the bottom. On this slide, we're seeing a laminar flow hood. This is another way that filtration is used. In this case, we have special filters. They're called HEPA filters that are installed in this hood, and then basically they are passing air through those filters all of the time and ensuring that the air inside the hood is sterile. So this provides a sterile environment for doing a variety of different types of research and manipulations inside of. Sometimes we actually have a door that's pulled down in front as well to keep you away from the samples even more. Okay, so that's the laminar flow hood. Again, it's using filtration, very small holes and some type of filter, and then we're forcing the sample through that filter. Membrane filters, they have all different types of sizes. So this right here gives you an idea of how small the pore size has to be to filter out different microbes. You can see that when we get down to the smallest viruses, they're really hard to filter out because you can see that they can be as small as 0.01 microns or micrometers. So that would be the hardest thing to filter out. If we're talking about protozoa, fungi, algae, yeast, those are going to be on the larger end of things. One of the reasons why they're on the larger ends of things is that those are eukaryotic cells. Okay, but those are going to be able to be filtered out quite easily. Your viruses in general are fairly easy to filter out, but once you get down to the bottom of this, again, tiny viruses, some of your bacteria, they're going to be much more difficult to eliminate just using filtration methods. Osmotic pressure is also one way that we can control microbe growth. This is going to be using high concentrations of salt and sugar and applying that on the outside of an organism. And what it ultimately does is it causes the water to want to move out of the cell because we are really creating, if we put it right here, a hypertonic environment. And when we have a hypertonic environment, the water wants to move out of the cell. And so when it moves out, if too much water moves out, the cell is ultimately going to die. This inhibits the metabolism because water is so important for overall metabolic processes. In general, this is going to be overall less effective for fungi than it is for other organisms. Okay, but it can be very effective when we're talking about trying to limit the overall growth of different protozoa, maybe different bacterial cells. Radiation is another way that we can control some growth. There are different types of radiation, but this can be either high-speed subatomic particles or electromagnetic energy. And when these types of energy actually strike bacterial cells and the molecules within them, they can actually result in a lot of damage. Ionizing radiation is going to be radiation that contains enough energy to eject electrons from the atoms. So when electrons are ejected, it creates ions, which will be charged atoms. Those ions that are created, they can be very damaging for various molecules inside the cell. This would be different proteins, could be denatured. We could have DNA that is ultimately damaged and not going to be very readable for the cell. We could have mutations that are resulting from this. If you look at the strawberries in this picture right here, it's showing you some that have been irradiated and some that have not been irradiated. And you can see that if you take these and you leave the samples for a few days, there is a huge difference in the overall microbe growth on the ones that did not have any particular treatment. And again, that's because when we shoot that ionizing radiation at the strawberries, we are killing, damaging the microbes that are there on the surface. Another type of radiation is non-ionizing radiation. These are going to use much lower amounts of energy. So when they use lower amounts of energy, we're not going to force electrons out of orbit. We're not going to create ions. And what these are capable of doing is that these can affect DNA structure. And if we're affecting DNA structure, we can potentially be affecting the ability for microbes to grow. So treatment with non-ionizing radiation does have some potential benefits as well. If we look at some chemical methods that can be used to control microbe growth, these would be chemicals that can adversely affect cell walls, membranes, proteins, and then also DNA. So they can damage all of those things and in the end affect the overall metabolism, reproduction, growth of the microbes. There is a long list of chemicals that we could include here and go through. So we'll just talk about some of kind of the biggest groups, most popular groups that are used. Phenol and then derivatives of that, which we call phenolics, are ones that are quite popular as being used for chemical control of microbial growth. You can see that phenol is basically a 6-membered ring. You can see it in the first part of the image here. And then you've got other kind of spins on phenol. These are going to be able to cause protein denaturation, so unfolding of proteins. That would be enzymes that you have inside of your cells, maybe some transport molecules, communication molecules. So that would definitely affect the overall metabolism of the cell and its ability to reproduce. These also can affect the overall cell membranes. So again, they would, in that case, be affecting the control of what gets to go in and what gets to go out, which would result in probably decreasing the overall survivability of the cells. Another group are the alcohols. Alcohols, here's two of the most commonly used ones. We have isopropanol, which is a 3-carbon alcohol, and then we also have ethanol, which is a 2-carbon alcohol. These are able to cause protein denaturation, disruption of some cell membranes. Many times when we're getting ready to, say, get a shot, the area that's going to receive the shot is going to just get swabbed with some alcohol right there. And again, that's going to cause some denaturation and disruption of cell membranes for any microbes that happen to be on the surface of our skin. Halogens are going to be highly reactive, non-metallic chemical elements. Iodine, chlorine, bromine, fluorine, these are all examples of those. So those are highly reactive. Oxidizing agents, can be used. These release oxygen radicals, which those oxygen radicals, they can cause denaturation of a variety of enzymes. So that means that they can turn off, inhibit a lot of different microbe reactions inside. And these are particularly effective against anaerobic organisms. And one of the reasons for that is that anaerobic organisms, they don't grow in the presence of oxygen because they are especially susceptible to oxidizing agents. When we're looking at things that are typically grown in the presence of oxygen, they do have some processes in place already to help them deal with oxidizing agents. So it's going to be a little bit harder to use those oxidizing agents with them. Surfactants are capable of reducing the surface tension of solvents, and if we reduce the surface tension of solvents, this is going to allow for more effective dissolving of solutes. This is what soaps and detergents do. And what you're seeing here is just two examples of what we call surfactants. Heavy metals are another one that can be used for chemically controlling microbes. These are going to combine with sulfur atoms. The sulfur atoms are found in cysteine, which is an amino acid, which would be incorporated into a lot of proteins. So you have a lot of proteins that have the cysteine, and the heavy metal is going to cause those particular proteins to not be functional anymore. And you can see in this image here that when you put the heavy metal there, you have this ring around it where you do not have any microbial growth taking place because of the protein denaturation that would be occurring. Aldehydes are chemicals that are capable of cross-linking with a variety of functional groups, and when they cross-link and interact with these other functional groups, they cause protein denaturation and nucleic acid inactivation. Here's just two examples of aldehydes. We have glutaraldehyde and then formaldehyde, which is probably one that you've heard of before. If we did want to evaluate the overall effectiveness of disinfectants and antiseptics, we do have a few different techniques that we use for that. The phenol coefficient is a test that is going to compare the effectiveness of our chemical of choice to the effectiveness of phenol, which was one of the chemicals that we just spoke about. If something has a value greater than 1, what that means is that it is more effective in its overall kill power than phenol would be. If we have something that has a phenol coefficient of less than 1, that means that it's less effective than phenol. So we can get an overall idea of how good this is going to actually work for us. We have a use dilution test. That's another way to evaluate a particular chemical. In this test, we're going to test different concentrations of our select chemical, and we're going to test its ability to kill an example bacteria. The Kelsey-Sykes capacity test is going to test for the growth of microbes after incubation with our chemical. And then another one is the in-use test, which is going to compare selective swabs of untreated and treated areas for microbial growth. So overall, what we're seeing with these evaluation tests is that we have to take our chemical and we have to compare with different microbes whether it's able to kill them. And then once we establish whether it's able to kill them or not, we want to establish what the best concentration of the chemical to use happens to be. And that lowest concentration is going to really be the ideal concentration for us, because we don't want to cause damage that we don't need to cause to the test site that we're treating.